Okay, thank you. So I'll be talking about the complement cascade in schizophrenia today. So I don't have any disclosures. So the complement cascade consists of a family of about 60 or so circulating proteins that form part of the immune system and whose role it is to literally complement the activity of antibodies and phagocytic cells. And these proteins act in a sequential-like or a cascade-like manner to exert their effects. And so they have a variety of functions, including the formation of the membrane attack complex leading to cell lysis, direct phagocytosis, as well as attracting macrophages and neutrophils to the sites of inflammation. So the complement cascade in psychosis has received increasing attention of late, mainly due to the recent genetic findings published in Nature by Seeker and colleagues that establish a link between the structural variation in the C4 gene with increased C4R RNA brain expression, as well as increased risk for developing schizophrenia. So whilst these findings generated a lot of excitement in the field, there still remains a lot to be explored regarding the complement cascade in psychosis and schizophrenia in general. So what do we know about the complement cascade in psychosis, and what remains to be explored? So of the studies that have examined complement proteins in the peripheral blood in schizophrenia, increased levels have been detected in some, but not all studies. And some of these discrepancies could be due to patients at different stages of illness, but it still remains to be seen whether these proteins vary across stages of psychotic illness. And of studies that have examined the relationship between protein levels and symptoms, these have been contradictory. So it still remains unclear whether any variation in complement protein levels have any clinical implications. And so we attempted to explore this link by examining the pattern of complement proteins with the pattern of clinical symptoms. And lastly, the complement factors C1q, C3 and C4 have been implicated in synaptic pruning in rodent models. However, this has yet to be largely translated into living humans. And so one way that we could indirectly explore the relationship or synaptic pruning processes is to examine cortical thickness on MRI. However, it remains to be explored whether complement proteins are associated with cortical thickness in living humans and whether this might differ in patients with psychosis. So this brings me to the aims of the current study. So we aim to characterise the complement factors C1q, C3 and C4 levels in patients at different stages of psychosis or schizophrenia, specifically those at clinical high risk for developing psychosis, those experiencing their first episode of psychosis, and those with chronic established schizophrenia. Our first aim was to determine whether these protein levels were altered in schizophrenia in comparison to their age-matched healthy control groups. Our second aim was to determine whether there was an association between variation in complement protein levels with variation with clinical symptoms. And our third aim was to determine whether there was an association between complement protein levels and cortical thickness. So here is just the sample characteristics. So this study consisted of 187 participants, again consisting of those at UHR for psychosis, FEP and chronic schizophrenia. And we had 2 healthy control groups that were age matched to their respective patient groups, so a younger and older healthy control group. And we measured key complement proteins, C1q, C3 and C4 in the serum. Using the human complement panel 2 multiplex assay. These were taken on fasting blood samples taken in the morning. The patient groups were compared relative to their age and gender matched healthy controls using permutation-based 2-sample t-tests, and the comparisons were corrected using the false discovery rate procedure. And we examined the relationship between the complement proteins with symptoms and cortical thickness values using canonical correlation analysis. So just moving to the results, for individuals at risk for psychosis, we found that the C3 and C4 protein levels were significantly increased in comparison to their age-matched controls, whilst the C1q protein remained unchanged. And likewise, in the first episode psychosis, the C3 and C4 protein was elevated whilst the C1q remained unchanged in comparison to their age-matched counterparts. Whereas in the chronic schizophrenia patients, only the C4 protein was significantly elevated, whilst the C1q and C3 protein remain unchanged. So it also examines the effect of potential confounds on these results. So we didn't find any significant difference in complement proteins between current smokers and current cannabis users with the non-users. We also didn't find a significant association between the protein levels with CPZ equivalent dose. However, we did find a positive correlation between the protein levels and body mass index in the entire sample. So we treated this as a nuisance covariate. And when we did control for BMI, the group differences in protein levels held for the UHR and the chronic schizophrenia groups. However, they were substantially reduced in the first episode psychosis group, and they were no longer significant. So in other words, the elevations in C3 and C4 protein levels we saw in the FEP patients were likely partly explained by increased BMI levels. So we used canonical correlation analysis to identify any potential latent relations between the set of complement proteins with the set of symptom scores and also regional thickness values. So we did not compare each complement protein with each symptom or thickness item individually, which is a process that would mandate a large number of comparisons and would reduce statistical power. So rather we used this multivariate approach because we thought it might be potentially more sensitive to uncovering more complex or nuanced relationships amongst the set of variables. So using this CCA approach for the set of symptom items, we found one significant mode or component in the patient group. So I'll just talk you through this graph. So for the sets of symptom items, the symptoms that significantly contributed to this mode were the negative and the positive symptoms. And for the set of complement proteins, the proteins that significantly contributed to this mode were the C3, which was a decreased weight, and the C4, which had an increased weight proteins, whereas the C1q protein and general symptoms did not significantly contribute to this mode or this relationship. So this mode indicated that decreased C3 protein levels together with increased C4 protein levels were associated with increased negative and positive symptoms. So when we examined the relationship with the proteins with each symptom item individually, we didn't find a significant association. So this led us to believe that it might have been the difference or the imbalance between C3 and C4 levels that was associated with the increases in positive and negative symptoms. So to test this hypothesis, we calculated the ratio between C4 and C3 protein levels, and we tested the correlation between this ratio with the symptoms. And we found a positive relationship between the ratio and with positive and two categories of negative symptoms, but not general psychopathology. And these correlations were significant after removing a couple of outliers in this data, although the correlations are quite weak. So again, it might suggest that the imbalance between C4 and C3 is driving the association with symptom severity. So what about any relationship with cortical thickness? So these results are quite initial and preliminary at this stage. So again, we used a multivariate approach, these canonical correlation analyses, to test whether the sets of complement proteins were associated with a set of regional thickness values. And the regional thickness values were taken from free surface software using the Deskin-Kiliani atlas. And we performed this CCA analysis in the combined patients of patients and controls, and we performed this separately for the left and the right hemisphere. And we didn't find any significant correlation in the right hemisphere. However, we did find one significant mode or component in the left hemisphere. And so this text sort of graphic here just explains or details the nature of this relationship when you drill down further. And so here The proteins and the regions that are presented in the same colour, so blue, they indicate that there is a positive relationship between the items, whilst the proteins and the regions presented in different colours indicate that there is a negative relationship between the items. The size of the text demonstrates the strength of the association, and the text that is in italics indicates the associations that are significant. So here, this just shows that C4 as well as C3 proteins were negatively associated with thickness in the left insula. So in other words, that individuals with a higher C4 and C3 proteins had a thinner insula in the left hemisphere. So if we look at this just via some univariate correlations, we find that again in the whole sample, we see that we do see a negative correlation between complement C4 as well as C3 with the thickness of the insula. Interestingly, the plots on the right, when we stratify by patient group, we actually find that it's the healthy controls that are actually driving this association. So here, the controls are illustrated in green and the patients in blue, and we see a negative relationship between C4 and marginally C3 with the left insula in the controls, but there's no evidence of this relationship in patients. So it's, yes, we're just working out what that might mean. So just to summarise these findings, we found increased complement proteins in peripheral blood in people with schizophrenia, especially those at the early stages of the illness or those at clinical high risk for developing the illness. We also found a molecular pattern characterised by decreased C3 together with increased C4 that was associated with increases in positive and negative symptom severity. We found that increased C4 and C3 levels were associated with a thinner insular cortex, but this was primarily driven by the healthy controls. And this might provide some initial or partial support to a complement-mediated synaptic pruning hypothesis of psychosis. However, again, it's very early days, and the nature of this relationship in those with actual psychosis remains to be kind of explored further. So just lastly, in terms of some future directions, I think it's important for us to measure a larger number of complement factors. Of course, this was only measuring 3 complement proteins within at the cascade. Again, we would like to measure complement proteins in both blood and the CSF to get a more direct marker of what might be occurring in the CNS. The sample of the UHR patients was very, very small, so of course those findings need to be interpreted with caution, and so we'd definitely like to examine the complement cascade in a larger number of individuals at risk for psychosis, and would also like to further examine the relationship between the complement factors with longitudinal changes in cortical thickness and symptoms, as well as other measures such as, for example, cognition. And I would just like to acknowledge some other investigators and authors that contributed to this work, particularly Liliana Laskaris, Andrew Zaleski, and Christos Pantelis, and Cindy Wycott as well. Thanks.
Thank you. Very interesting talk. We have time for some questions. Yes.
Thank you. Very interesting study. Obviously, lots of data, and exploring what it might mean. Wouldn't one approach for the neuroimaging element be to look at pairwise differences between thickness between cases and controls, and then look at how that correlates with complement? Because having different regression lines for the controls and the cases is pretty hard to interpret.
Yes, it is at this stage. So one way with that we were thinking of exploring this further was to be looking at, yes, the between group differences, and then exactly putting that into the canonical correlation analysis. Yeah, it's a good idea.
Thank you for a great talk. Just one question. Did you by any chance look at IL-6, TNF-alpha, CRP in terms of potential correlations with the complement proteins?
So we have measured some other cytokines, and we didn't find any elevations in cytokines, as well as, I think, CRP only in the chronic schizophrenia group. I believe CRP and the complement factors were correlated. I'd have to double check that. But we didn't find any changes in actual other pro-inflammatory cytokines in this group, as well as in a subsample of this group, there were no changes in TSPO levels as measured from PET as well.
But CRP was correlated with the complement?
I believe so. I have to double check. Yeah, I could get back to you.
Thank you.
The fact that you have correlation with the ratio, but C3, C4, is it, so from what we know from the role of C3 and C4, is it make it sense? And also with the thinning of the cortex with C3 and C4, and only in insula and the left hemisphere. Could you comment on that in the biological role, what we know now from C3, C4?
So yes, of course, so not all the findings are reconciling, and it's not a neat picture at this stage, but it never is. In terms of the ratio, look, if you have any insights yourself, I'd love to hear them, but perhaps because it's in a cascade-like manner, you might be finding that C3 could be downregulating due to increased C4 levels. That might be a possibility, given that it works within a cascade. The findings specifically to the insula, I mean, again, this is taking a whole brain approach, so we haven't actually targeted particular regions. It is a region that is changing in the early stages of schizophrenia and psychosis and over the course from high risk to transition. So it is implicated in the early phases of the illness, but I can't really comment further in terms of biological mechanisms.
Okay. Thank you very much.
Thank you.